Hi everyone, welcome to OceanX Live. Ramadan Mubarak and happy early Earth Day. My name is Shannon Forey. I'm part of the digital team here at OceanX, and I'm thrilled to welcome you to tonight's live classroom. OceanX is on a mission to explore the ocean and bring it back to the world. We are currently underway on the Red Sea Decade Expedition in partnership with the National Center for Wildlife of Saudi Arabia. This expedition is all about building a complete scientific picture of the Red Sea, from the depths to the shallows, north to south, microbes to whales. Our speaker tonight is Sofia Frappi. She's a PhD student specializing in Elasmobranch Conservation at King Abdullah University of Science and Technology. We'd also like to give a big thank you to our guests on the call from the biology department from the Haran School. Hello and welcome. One final note, we'll have time at the end for questions. So during the presentation, please be thinking of those and getting ready for our Q&A session. And now let's get right into it. eDNA and elasmobranches with Sofia.
Hello, everyone, and welcome to this OceanXplorer class on elasmobranchs and environmental DNA research. During this presentation, I will give you a brief resume on what elasmobranchs are, what environmental DNA is, and which kind of research are we conducting on board on OceanX. So, it's starting very badly. So, let's start with the elasmobranchs, and let's say that they are among the most diverse and ancient marine vertebrates on our planet. They are part of the phylum Chordata in the class Chondroichthys that encompasses all the cartilaginous fishes, and it opposes to the class Osteichthyes, in which we find all the bony fish. And then we found in the class Chondroichthys a subclass Elasmobranchi, that is the one that we're going to investigate, and another class that is the subclass Holocephali. This is just for your information. I'm not gonna talk about them during this presentation, but just for your information, these are all the fishes that we define as chimeras. So they are still cartilaginous. And I don't know if you ever heard about rabbit fish, elephant fish, but anyway, very weird deep sea creature. But in our case, we're gonna focus on the subclass Elasmobranchi, and that itself is divided into superorder. One are the Selachimorpha, as we know, sharks. And then the other superorder is the superorder Batoidea. And in this superorder, we find all the other cartilaginous fishes apart from sharks. So they have various characteristics that communate them. And one of the most evident is the dorsoventral flattened shape they have. So we find, for instance, stingrays and sawfish here. While, as you can see, we have a tiger shark that can be can be clearly part of the superorder Selachimorpha. So let's go a little bit deeper into details when we talk about sharks. This superorder comprehends 8 different orders of sharks that in turn, they account for all the existent species of sharks in our planet. So we found Exanchiformes that have as a representative the well-known frillet sharks and cow sharks, and especially the frillet sharks are all the sharks defined like living fossils because they have characteristics that are clearly primitive. So basically they didn't change much during time. Then we find in the order Squaliformes, the so-called dogfish sharks. In this case, this one is the Squalus acanthias, or spiny dogfish. And it's called like this because he has a little spine in front of the dorsal fin. Then we find the Prisioforiformes, that are the sow sharks. And the etymology of the word Prisioforiformes means literally bringer of the sword. So in fact, as you can see, they have this rostrum that is highly modified with a lot of teeth. And we have to pay attention when we talk about sow shark because they are not sawfish, they are very different. And the main difference is due to how do we distinguish them. Because of the position of the gills. In the sharks, the gills will always be lateral, while in the rays and also in the soul shark, that is a batoidea, will be underneath their body. So the other order is Squatiniformes. It comprehends all the angel sharks. This one is Squatina squatina. Then the Pterodontiformes, the bullhead sharks. This one is the Port Jackson shark. And then we have the order Lamniformes that comprehends some of the most famous species of shark. This one is the Isurus oxyrhynchus, the mako shark. And in this order, we also have the most famous shark, the Carcharodon carcharias, the great white shark. Then let's go with the Orectolobiformes, or also called carpet sharks. So we have a whale shark that is well known, but then maybe the Wobbegong shark, the second one, is not so well known, but then you get the idea why the common name of this order of shark are carpet sharks, right? And then the Carchariniformes that encompasses for a large number of species, and they are generally called ground sharks. This one is one of the most famous, is the black tip reef shark, the Carcharinus melanopterus, and it's usually found around reefs. So why are these animals so important? Why do we want to protect them? We have to keep in mind that if we want healthy oceans, we also need healthy population out there. And healthy population depends strictly on the presence of apex predator in their environment. So apex predator exercise what we call a top-down control on the ecosystems. If we remove them, then we will cause something called trophic cascade effect. And this will lead to bad consequences for the environment. Sometimes we will have the shift of entire ecosystem, and so the transformation of the environment in something different. In general, sharks keep in check the overall health of fisheries, and I love to call them the quality check team of the ocean, and they maintain the biodiversity within ecosystem. You have to imagine that they have been here for more than 400 million of years. And they have been shaping indirectly the habitat structure in which they were living, as well as the ecosystem productivity. Someone called them the architect of the oceans. Moreover, the species, so sharks, and also elasmobranchs in general, because also rays are very important from this point of view, they play a fundamental role in the tourism, especially in some part of the world. Then when we talk about their importance, we also have to talk about all the threats that they are facing, especially nowadays. We know that they are undergoing overexploitation and unregulated fisheries. So, for instance, have you ever heard about the shark finning practice? Of course you have. And basically, the shark fins have a high price on the market, especially on the Asian market, because they are used as ingredients for a soup that has a socio-economical value. That means that this soup is not particularly good. In fact, it's full of very bad substances for our body, for instance, high in mercury. But still, because shark's meat is full of pollutants, full of heavy metal. But anyway, this soup has an importance from an economic, socio-economic and socio-cultural point of view, because only healthy family can allow to buy shark's fins to make this soup, and they will serve it during a business gathering or marriages, and so they will show off their health and their social status. Then we have the bycatch that for sharks in some part of the world goes up to more than the 90% of the total catch of the fisheries operation, especially with some fisheries device like the long line fishing. And then we have the ghost fishing that is very sad. Maybe it's not the most known, but you know that sometimes fishing tools get lost in the ocean, or the nets got tangled somewhere on the bottom of the ocean, and the fisheries operators leave them there. It's too costly to retrieve them, so they keep fishing underwater, and they create these long areas and huge areas in which you have a lot of dead animals entangled in this ghost net underwater. Then, of course, there are other motivations for which this animal can become target fisheries. One of them is the use of one compound in their liver oil, that is squalene, and is used in the cosmetic industry. So how many sharks do we lose per year in total? From 70 to 100 millions. And it's a big number. And what is the situation here in the Red Sea? Well, the biggest problem is that we lack extensive information on the abundance and diversity of elasmobranchs in this region. And even if there is a ban on shark fisheries, the unregulated fishery go on because it's very, very hard to control fishery operation, especially out at sea. So what is the role of science in all this? We want to get our data because this is at the base of the creation of management conservation plan for these endangered species. And when we are able to gather extensive data, ideally that must be collected over a broad geographic ranges and over a long period of time. That's why being here on OceanXplorer is very important. Now we are able to go all around the coast of Saudi Arabia, gathering a lot of information. So the results can be used to implement policies for the protection of endangered species. And this can be done at a worldwide level. For instance, in the past, we saw the establishment of CITES agreement, that is the Convention on International Trade of Endangered Species, and it applies both for fauna and flora. And then right now, it encompasses something like 30 species of rays and sharks. So all the traffic, all the trades of these species and their related products is strictly highly regulated. And then we have also the establishment of specific policies in areas of the world, in different regions. And one example of this could be the establishment of shark sanctuaries, in which sharks, in general elasmobranch, but in most cases it applies especially to sharks, they are protected by law. So every activity concerning their fisheries or any kind of disturbance to these animals is prohibited. And the first establishment of one of these areas was in Palau in 2009. In total, now we have 17 shark sanctuaries in the world, and the last one was established in Samoa in 2018. But why we talk about environmental DNA? First, we need to understand which kind of monitoring techniques have been used for all this time before the invention of new technological tools. So the point is that the monitoring of this population comes with many problems at the base. And among these problems, there is the low abundance of these predators. Some of them move a lot around wide geographic areas and even over long distances. So they're very hard to spot and study. And then there is their elusive behavior. So many studies until now have been conducted using standard technique like the telemetry, so tagging shark and observing by remote their movements, visual observation, both from vessels or scuba diving, the use of the BRAF, that I don't know if you have ever heard of it, but is a baited remote underwater videos. Basically, you have this support in which you mount a bait, and then you put on the camera and put it on recording for some times. It depends on the battery of the camera. And then you deploy it somewhere and you wait to see which kind of footages you get. And it's usually for the study of elasmobranch it's quite famous, but still has a lot of limitation. For instance, you cannot deploy this if you have strong currents. Anyway, other techniques have been the UAV, that is the use of drones, so observing from the surface. Then if you want to have a general picture of the biodiversity of elasmobranch in an area, you can rely on fisheries. So you go to fish market and you do surveys. This has been done here in the Red Sea. And then you go and rely on fisheries bycatch and on counting of strandings. But these methods have problems, of course. They are, for instance, time-consuming, expensive, and less successful in detecting rare and elusive species. Sometimes it is also difficult to correctly identify some cryptic species. Cryptic species mean that they look alike from the surface, but then they are genetically different. And sometimes it's also hard to distinguish between a juvenile and an adult stage in some animals. So another problem is also related to the non-standardized technique that get applied, and also to the invasive nature of some of these approaches. And these increase the risk both for the animal and for the researcher involved in the process. Let me give you an example. So this guy is tagging a shark, is not hurting him, it's just the normal tagging technique. The point is that this kind of method will create a lot of stress for the animal. So even if it gets safely released in the water, it doesn't mean that the big shock he withstanded will leave him unharmed. Sometimes they also die due to the stress they had to go through. Professor Carlos Duarte is collaborating with this association, Beneath the Wave is an NGO that works in the Bahamas, and they do tagging of sharks. And actually, these guys developed a technique to minimize the stress on the animal while doing the tagging. So they leave the animal in the water, they minimize the interaction with it, and they try to do a lot of analysis in like 5 minutes. They take samples, measurement, they put tags on it, but still, like, you have to run, because in the end, if you don't do this, you will cause risk for the animal and also for yourself. If the animal gets agitated, there is always a risk involved in such operation. So my question is, at the end of this speech, can you find a fish in a vial? Let's get to the meaning of environmental DNA. Today, we have a modern and effective, non-invasive, and less time and labor-consuming tool for elasmobranch monitoring, but in general for biodiversity monitoring. And then this method uses the DNA that is naturally released by the organisms that live in a certain environment. This DNA is called environmental DNA. So this analysis is possible thanks to the development of specific technologies like the next generation sequencing, which allows for extensive biodiversity surveys with limited efforts and costs. Environmental DNA has been applied to monitor ecosystem, and you can target single individuals or multiple species, all at the same time. And you can use different kind of samples that can be fresh water, saltwater, sediment, and so on. And also you can target different periods of time. For instance, there is modern and ancient eDNA. But let's talk about the principal character of this presentation, to the protagonist of this talk. That is the deoxyribonucleic acid, the DNA. So, as you can imagine, the DNA is like our instruction booklet. It contains all the information that makes us what we are, and everyone has little differences in their DNA, otherwise we would be clones of each other. And As you see, is double-stranded, his structure is double-stranded, and it has this helicodial shape. And the basic units of the DNA are what we call nucleotides. The nucleotides are made by three different parts. One phosphate group, one sugar, that in this case is the deoxyribosome, and one base. We have four different bases in the DNA. We have adenine, thymine, guanine, and cytosine. Adenine and thymine will always bound together, as well as guanine and cytosine will do. So let's say that the backbone of the DNA is constituted by the sugar and the phosphate group. But then the double strand of the DNA is maintained through hydrogen bonds between the different bases. Now, I want to point out your attention to two processes, the transcription and translation, because this will give us a better idea about the functioning of the DNA. So basically, you have to imagine that the bases on the single strand of the DNA act as a code. So three letters together forms codon, and each codon will code for specific amino acid. The amino acid are the units, fundamental units for the construction of proteins, and proteins are essential for our daily function. So you will have the need to create proteins, and this is why we have the process called transcription and the process called translation. So basically, during the transcription event, you will have an enzyme that is called RNA polymerase that will attach to a single strand of the DNA and will mediate this process of RNA transcription. So you will have a messenger RNA that will leave the nucleus and will go in the cytoplasm of the cell, where it will encounter a ribosome. And here there will be the translation process taking place. And in the translation process, again, you will have the transformation of this transcript that you obtain from the DNA in real proteins. So why is this important? You will see later on. This is the most, let's say, a more reliable reconstruction of how the DNA will look like, of course, without small letters, but this is how it is. And now I want to ask you if you know what forensic analyses are. Of course you know, because if you at least saw once in your life CSI or any kind of crime TV show, you will know that there are these science teams, the scientists that will come that will go in the crime scene, and they will start taking samples. They will search for sample, biological traces of the killer, and so they will take the samples, these biological traces, back to the lab. They will extract the DNA, and then when they have the sequence of the DNA extracted, they will go to the police archive, in which during time a lot of criminal DNA got accumulated, and they will try to find a match with what they have and what's inside the archive. If they find a match, maybe they have been lucky with this investigation. But why I'm telling you this? Because I ask you, is this possible to apply a similar approach to study biodiversity? So let's say you have a biological samples, because you can get it through sediment, water, whatever you want. And inside these samples, there are biological traces of the animal you want to study. It's not to a single individual, but to a single species, we can apply this approach. So we can extract the DNA that we have in our biological sample, and we can, in the end, have a sequence. And then we need the reference archive. And we have a reference archive in which, during time, a lot of sequence genetic code of a lot of animals that have been studied have been stored there. So we have a lot of sequences of studied animals. And we can go there with our sequence and see if we can find a match. And then this is how you know what you have in your samples. But this is a very simplified explanation. I can go further in detail talking about my own project. So I study elasmobranchs in the Red Sea. And on the OceanX, I study the elasmobranch samples that I will find in the deep ocean. So we go there and we take course. What does it mean? We have this beautiful remotely operated vehicle that is a ROV that is called Chimera, is the OceanX ROV. And what does it do? It goes deep down in the ocean, can get down to 3,000, even more. I think the deepest point we reached was 3,000 until now. I'm not sure though. Anyway, we go there, and as you see, he has a lot of tools, different tools for different sampling goals. In my case, we're gonna use the two, not two, we take more, but anyway, we're gonna use cores. And the metallic arm will go here, we'll take a handle of the core, push the core inside the ground, take it out, and with the specific mechanism you have inside the core, the sediment will not slip out, it will get stuck in there. And then it will put it back in the crane, and when it comes up, as scientists, as a nerdy researcher, we'll go there, take the course, and go back in the lab. And when we go back in the lab, we basically gather halicots from the sediments. We store them safely in the fridge until we are able to go through the next steps. For the next steps, we need a lab, because then we're gonna do the DNA extraction. Because as I told you, yes, the sediments is full of DNA, but the DNA is inside the cells. So you have to go through a chemically mediated process that is made of many steps in order to do a cell lysis and then transform what you have there into a vial with a droplet of material that is constituted only by environmental DNA. The third step is the polymerase chain reaction. And this process, we can define it as artificially inducted amplification of DNA. And what does it mean? You have to imagine that when we start in our samples that we extracted, we have DNA, but we have only few copies of our target DNA, of our DNA of interest. And these copies maybe are also degenerated a little bit. So what we need to do, we need to make copies of it. And this is possible because through this process, we will start with a few copies and we will end up with billions of copies. But how does it work? So this process that is widely used for many different purposes in the world has been invented in 1983, yes, by a biochemist, Katie Mullis. And the principle is that you have a short sequence of DNA called primers, that are the red ones here. And this primer will bind to specific portion of the genome of your target organism. This is important because this way you will filter out all the organisms you are not interested in. For me, I have primers that will attach only to the elasmobranchs DNA and will not amplify anything else. So you start with this, then of course you have the mediated process through an enzyme that is here are represented in orange. So these enzymes are polymerase, like the one that we saw before during the transcription and translation process in the DNA. And they actually serve to the same purpose. They will create double-stranded copies of DNA. But this kind of enzyme, they have been extracted from a bacteria that live in hot springs. I think is Thermophilus aquaticus, the name of the bacteria. And this is important because these enzymes are tolerant to thermal stress. And this is the most important thing during this process, because we're going to go through many changes in temperature and many thermal cycles. So if the enzyme die, the process stops. So this kind of enzymes, they will be resistant to everything that we will throw toward them. Anyway, how does the process work? So you have to imagine that you start, then your solution, you have the target DNA that is still double-stranded, the enzymes, the primers, and the nucleotides that are the basic units for the formation of the strand of DNA, as I told you before. So we have different cycles, we have different steps during these cycles. The first is the denaturation. As you see, you will heat up the DNA until almost 100 degrees. And what this will cause? It will cause a disruption of the hydrogen bonds that keeps together the two strands of DNA. And this will open up. And in the next step, the annealing, in which we will lower the temperature, you will have the attachment of the primer to the single strand of DNA, and then the attachment and the positioning of the polymerase enzyme, that in the next step, that in which you will heat up again the temperature, you will mediate the elongation process. So in the end, what you have are copies of your original target DNA. And then when you repeat the cycle again and again and again, you will end up with billions of copies of this DNA, and you will be able to go through the next step that comprehends the next generation sequencing and DNA metabarcoding analysis. So we have the machine that will tell us, so we will start with the amplified DNA. The machine will read this for us. It will sequence this amplified DNA. And then it will do a massive sequencing of all the amplified DNA that we had at the end of the PCR. And then when the sequencing is done, we will have reference databases that have inside all the known organism genome that has already been sequenced during time up until now. And then we will try to assign each sequence that we find in our data, in our environmental samples, to a known species. So it is, after this, you go through your bioinformatic analysis, and you go on reading your results and analyzing them. So to resume, just a very fast resume and easy peasy. From right to left, we start with the eDNA that gets accumulated in the sediment during time. We take the core, we scrap the surface layer of the core, and we bring it into the lab, and we do the DNA extraction. Then we go through the PCR, polymerase chain reaction step. And when we add our amplified DNA, we go through sequencing and bioinformatic data analysis. It looks very easy, no? In the end, we will have a list of animals, a list of target organisms that we wanted. It's not so easy. This process can be full of errors. In each step, you can commit errors. Sometimes it's just a casualty. So during the extraction, during the PCR, during the sequencing. And then the contamination is one of the biggest problems of this kind of study. You can imagine that just breathing on your samples will put your DNA inside that sample. So all the environmental DNA studies are full of scientists' DNA. And to just give you an idea of how sensitive is this method, when I was studying for my master, I was filtering water in a kitchen because that was the only place available I had. So I was sterilizing everything, covering everything in aluminum foil, putting sterile things everywhere. And then just the day before, my friend was eating a sandwich with the turkey inside, and then I found it. I found turkey in my analysis, and for sure I wasn't targeting that. Anyway, this is very, very sensitive method. But we don't stop just here, because we don't only try to do a geographical biodiversity assessment in respect to elasmobranch. We also try to study changes in their biodiversity during time. How is this possible? Well, you know that isotopes have been used until now to see the sedimentation rate and accumulation rate of sediments. So to date specific layer in a sediment core. And this is what we are gonna do as well, using as a isotope Li210. We are collecting cores for this specific analysis, each leg of the expedition. We slice them down in 1 centimeter slices until 20 centimeter in depth. And then from each of the slices, we take an aliquot of environmental DNA. And when we finish with the isotope analysis, we can say, we can tell the age of each layer. And then studying the data that we gather from each of this layer, each one of this layer, we will be able to investigate biodiversity changes during time. That's our aim. Let's see, that's our goal. Let's see how it plays out. So now I'm gonna give you some example of practical application of the environmental DNA method. So this study dates back to 2018 and has been run in New Caledonia. The researchers were trying to use different methods to see the potential of environmental DNA as a method to study sharks. So here you can see that there are 3 different methods that have been used. The red one is the samples they gathered with eDNA. Total number of samples, 22. And then the total number of species they found is 13. UVC is the underwater visual census, so I believe they were doing it scuba diving, and we do more than almost 3,000 dive, they saw 9 species of shark in total. And then with the BRAF that I told you before, with 385 dives of the BRAF, they saw 9 species of sharks. So this is not, I'm not saying that eDNA is better, and not this study is not saying that the eDNA is better than the other methods, but it is very important to note the small amount of effort in respect to the other methods and the number of species they were able to assess. Of course, the eDNA didn't catch the entire biodiversity. That's why it's important to always merge different methods, use different methods together to have a broader view. But this is just to give you an idea of how powerful can be this method. Another case in which using eDNA made a difference was this study in 2021 in Guam, Micronesia, in which for 50 years there was no scientific evidence of the presence of scalloped hammerhead sharks in the area. And then they went there, they did an environmental DNA assessment, and they found it. And this is very important because it's a critically endangered species. So this study can be used to push through better conservation measures for this animal. And then another interesting example is this one. It's a study that has been run in Singapore in 2021. And Singapore is highly urbanized, the water is turbid, so to study elusive species like elasmobranchs, it's very hard. Here in this case, the researcher used 2 different primers, the one in blue and the one in red in the picture, as you can see, but both of them are for environmental DNA studies. And then you have inside the black circle, the museum specimen, so the specimen that have been accumulated in the museum during time, and the green line represent the visual survey. So as you can see, the environmental DNA was able to gather all the already known species to be there, but also 2 species that have not even been assessed through. They're not even part to the museum collection, let's say. So the potential for a complex and more complete overview on the biodiversity of this hard to study environment, because this is urban areas, murky water. This is not easy environment to work with. The environmental DNA in such cases can be very, very important. So, as I told you, combining different methods can give you satisfying results. So, that's why during this expedition, we decided also to gather all the footages and videos of sharks that we encounter while we go down with the ROV, when we go down with the subs. And then some of these footages are really, really precious because they come from a very difficult environment to study, that is the deep sea. And in the end, the possibility to have these footages and the environmental DNA data will allow me for a comparison and for a broader view on the general biodiversity in these areas. But what did we see until now? I had made a selection of some clips that I want to show you. Some of our operation, this was an ROV operation that was targeting sharks in the deep sea. We are at 2,000 meters of depth. You will see interesting behavior of a species of shark that it's not so easy to study because it lives in a very unexplored and hard to reach environment. This is Iago manensis. It's called the big high ham shark. It is very small because, as you can see in the bag, we have bait. These are small, not small, but bonito fishes. So they're not even so big, but the shark is very small. So the laser from point to point, we use them to measure things. They should be like 10 centimeters. So the shark is not even 40 centimeters, maybe 30. But it's interesting to witness this behavior, this predatory behavior, because it probably is the first time that it got witnessed like this. Here, we freed the bait to let the shark hit. And this was a big party for her because it's very rare to have this kind of buffet underwater, this deep. And so this shark probably, with the energy she gathered from this single meal, will go on for days. She was almost bursting. She has very big belly. But again, these images are very, very important because probably like they are unique. To be able to witness this kind of shark, and its behavior, it's amazing. So you saw, no, that was a target operation for shark. We had the bait, we were shaking the bait, opening, trying to attract predators. But sometimes you get footages of sharks that just get close to the ROV while it travels underwater. And it's not a clear footage. So that's why having the DNA can help us a lot in identifying the species. Anyway, for instance, this one. This is a big shark and is on one of the lateral cameras of the ROV. But it's very hard to try to identify. I can say it's a Carcariniformes, but then I can just do a best guess on the species. Or sometimes the view is a little bit more clear, even if we are still in the dark because 500 meters is pitch black. Sometimes they get close to the light, but they go very fast. So again, it's very hard to identify them. Maybe the DNA that we will find in the environment will help us. Sometimes it's way easier because sharks get attracted by lights and by electricity, and they are a little bit, they stuck around for a little bit. These are very clear images, so I have my idea on the species of shark that we have here. So yeah, in this case, we have an exhibitionist, and it was amazing because but usually they just pass by. So it's very difficult to say for sure which kind of species is that. That's why it's important to also compare the data that we have in the environmental DNA. So this is Mati, our lead scientist on OceanX, and she was preparing a bait because up until now, the video you saw were gathered with the ROV in the deep ocean. But now in this specific operation, we decided to go with the sub at 130 meters on top of a plateau to do again another baited dive and searching for sharks. So she was cutting bonitos again as a bait, and it says going up with the sub until the top of the plateau of this underwater mountain, and we were waiting there for 40 minutes, shaking the bait and with the light off, and then finally we got to see what I was hoping to see. That is a Sphyrna lewini, a scalloped hammerhead shark. You see the bait in the water. So this is a critically endangered shark. This was something I really wanted to witness here. And this is a clip that our media team on OceanX released, and it's amazing. So I want to share it with you guys, and I hope you enjoy as much as I was enjoying. At a certain point, my happy face pop out. And anyway, that was an amazing experience. So both of the sharks we saw two scalloped hammerhead sharks. They were both females. One was a young individual. So thank you for your attention. I hope you enjoyed this talk, and I hope now you're passionate about elasmobranchs and environmental DNA studies.
Thank you so much, Sofia, for sharing your work with us. If you're joining us on YouTube, feel free to put your questions in the YouTube chat. If you're joining us from the biology department on the Zoom call, please use the raise hand function on Zoom, and we will try to get to everyone's questions while we have some time with Sofia. Hi, Sofia.
Hi, hello everyone.
So I have a great kickoff question for you, one of my own. What is the trophic cascade, and what is an example of something that happens when sharks are removed from an ecosystem?
I give you a very simplified example. So usually it's not so easy. There are a lot of different factors in play, but let's say if you remove in one of the reef predator, that can be like a reef shark, then you get like all the parrotfish that predates on corals, or algae that overgrow corals, that will start eating everything, right? Without any kind of limitation. And then in the end, you will have a real serious damage for the corals. And if they cannot recover in time, first, they will get overgrown by like algal turf. And so they will not, it will be not so easy for them to like overcome this. And at the same time, also the parrotfish themselves, they will have no more food, no more resources. Okay, so they will in the end leave the area, and that reef that was beautiful and very active and functional will become overgrown by algae. It will just be a change, you know, there will be other organisms living there, but it's not going to be a reef as we know of. So it's a change. It's going to be a shift in the environment. This is one of the very bad consequences of having a cascade effect. It's like a chain effect. So one event leads to the other, and it's very hard to stop this.
Thank you. That's a really great answer. We have some questions coming in from our YouTube channel. We have a question from Carly. Is there an equivalent to eDNA on the land or in the air? Can you use this, for example, with birds?
Of course, a lot of studies have been done, even in the Amazon forest, and they are also using air. And one of the most studied communities of organisms using environmental DNA is bacteria. And it was like the first organism on which environmental DNA was applied.
Great, thank you. And we get this question all the time on our social media channels for our scientists. What is your favorite type of shark?
Mine is the Sphyrnamocaran, is a great hammerhead shark, and I got very lucky because the last year during our expedition in the Bahamas, the last day they were baiting the water trying to catch shark, and we catch a big, amazing, beautiful, great hammerhead shark. But they are so strong, they are so powerful, and they get so stressed. Like, can you imagine how powerful can be that animal, that apex predator? Then if you get them like all worked up, and they get very stressed, like they're gonna die out of stress. Now our cruise leader at the time on shark operation was Austin Gallagher, the owner of Beneath the Wave, that NGO we are collaborating with. He was like, we cannot keep this shark on, like, and we cannot work with him more than 3 minutes. 3 minutes. And then you have to finish all the operation before, and then release him. Otherwise, it can die, and it is incredibly delicate.
Oh, wow. So I think we have a question from our biology classroom, so I will let them ask.
Sure.
Hello, can you hear me?
We can. Welcome.
Well, my name is Majid Faraj. I'm an 11th grade student, and I have one question. That is, is collecting samples in the Red Sea easier than other places?
Sorry, say it again.
Is collecting samples in the Red Sea easier than other locations?
You mean to get the samples, if it's easier here?
Yes.
I would say what we are doing is not easy anywhere, because we are sampling the deep ocean. We go down to 2,000 meters, even more than 2,000 meters, and because of the pressure at that depth and many other factors, like it's very, very difficult and hard to reach there. But in fact, like environmental DNA is one of the few methods with which you can study biodiversity in this very challenging environment. So like what we are doing here, especially on this boat, like it's never easy. So it's great to have the chance to do it now. We also getting samples from shallower water, but we are not doing heat on this boat. We're doing it with another boat that is a Halazizi, is a partner boat that we are using to study shallow water environment. It's the same operation. We are doing it at the same time, and we're getting samples there. And I have to say, I would not say it's more difficult, but for sure it's more interesting here than in other places, because as you know, the Red Sea is full of biodiversity, full of endemism. It's an amazing place to study in terms of biodiversity.
Great, thank you so much, Sophia. Do we have another question from the class?
Yeah, hi. So my name is Abdullah Shoaibi. I'm from also an 11th grade student. I just wanted to ask, with all these big Red Sea mega projects, do you think it's possible for large-scale projects to avoid harming aquatic ecosystems?
I think it's always possible to try to minimize at least the impact. And science has always a dark side, because as you know, like even studying environmental DNA, we unfortunately use a lot of reuse of single-use tools in the lab, so you have to throw them away, but there is always a way to minimize it. And I think that the Giga project of the Red Sea, they are trying to find the most sustainable way to do things. So it's already like a good first step.
And Sophia, I have a quick question with that. And how, in studying places like the Red Sea, how can that help create places that are marine protected locations and things like that? Can you speak to how your science is helping to contribute to that?
Oh, well, for instance, like you can get a lot of data on, like in my case, elasmobranchs, they are very difficult to study. So like if you don't have extensive data on a population, you cannot say, let's establish a policy that will protect this animal here. For instance, let's create a marine protected area, or in case of sharks, a shark sanctuary. If you don't have data, you cannot do it. So in the Red Sea, you have an incredible varieties of sharks. And what we miss are the numbers and the distribution, you know, because without knowing the distribution and maybe also important areas for their life cycles, nurseries or feeding ground, then if you know this, you can try to push for conservation policies. You can do it here, you can do it in other areas of the world, but what I think is that the richness you have here is very high, and so it's worth to protect it.
That's great. Thank you, Sophia. We have another quick question from YouTube, and then we'll jump back to our wonderful classroom. We have a YouTube question about what happens when there's a new species or strand of DNA that cannot be identified. And that's from Sam on YouTube.
That's a very good question. So what you have at the end of your process, if you have this sequence that doesn't match with anything, there are two possibilities. You found a new species, and in that case, there's no way you're gonna know, because you know that this is unidentified, but you don't know what is it. Maybe you have like it will bound to not a specific animal, but a genus. Or I don't know, like Carcharodon, like, you know, it's a genus, it's not a species, but you know what we're talking about already. So you can distinguish is a shark, is a fish, is a, I don't know, a bird, you know. So you can already say this, but then if you cannot get to the species level, you either found a new species, that would be pretty cool, but you will never know. Or second, you found a species that is already known, but its genome is not in the reference archive. So to be in the reference archive, that genome has to be studied. So someone has to decide, let's study this animal, let's sequence all its genome, and let's deposit it. Sometimes there's not even the necessity of sequence all the genome of the animal. You can only sequence some portion, and then you just deposit that in this archive. And if you use a primer that attached to that, you will be able to find that animal. If you use a different primer, you will not be able to say, oh, it's that. So the point is many samples now that have been collected years ago, they are being rerun. So they are doing again the analysis, because now, in respect to 3 years ago, 4 years ago, we have more sequences deposited. So someone studied more animals in the meanwhile, more organisms, let's say. And so maybe if you rerun a sample that you took 2 years, 3 years ago, and you couldn't find a match, maybe today you find a match, because someone studied an animal and deposited the genome inside the archive.
Awesome.
I hope
so it
was.
So I want to make sure I get a couple more questions from our classroom since they're joining us. Anyone else from our biology class would like to ask a question to Sophia?
Go ahead.
How can you use engineering principles to maintain biodiversity in the Red Sea and improve our understanding?
Sorry, I can't hear very well. The audio is pretty. Can you repeat the question? Sorry.
How can you use engineering principles to maintain biodiversity in the Red Sea and improve our understanding of it?
Okay, how can you see it? How can you use it? How can you use this information? Right? So you can do
Sophia,
a map.
And how can you bring in engineering into that science as well?
And engineering. We were talking about many ideas about how you can actually apply different science, no? So it can be like interdisciplinary field. And for instance, you can build devices that will sample every time, maybe in different season, maybe because you know that that season is specifically important for that animal. So like you want to monitor the presence and the abundance of, let's say, a stock of fish that you know that is very, very important from an economical point of view. And you want to know where is the presence of that stock, maybe if there is a migration pattern, maybe if his presence will appear seasonally. So you want to study that, you want to keep in check that species. That's just an example, no? But that's what do you need is like the possibility to sample for extensive period of time and extensive areas. So you build devices that maybe can stay out at sea without even consuming fuel, so being completely green and sustainable, and then stay out there and sample for a long time. And then you go there, you retrieve your data. Once in a while, you can filter water, you know, and that would be like an interesting, for instance, engineering application to this. But then there are many other ideas, and we are open for ideas, guys. If you have one, you feel welcome to share it.
Great, and I want to squeeze in one more question from our wonderful class. So if anyone would like to ask our last question before we wrap up here.
Hi, I'm Jenna Patlani from grade 10, and I want to ask, do you plan to use eDNA in other fields, like for example in genetic engineering? And is it possible to study other species using the same method?
I mean, different species than elasmobranchs, you mean, in general. You can study all the organism you want, as long as their genome is deposited in the archives. Otherwise, we will not have a match. And I mean, environmental DNA can be applied to a lot of things. I don't know what you mean when you say like exactly genetic engineering, like if we can actually modify. That's what I don't understand what you mean by that. But I'm pretty sure, like if that is some practical application for that, we will find out later on. But yeah, of course, you have to think that as long as you have an environmental sample. It can be air, it can be water, it can be sediment, whatever you think of, any medium. So as long as you have that, and as long as you have a reference archive in which you can actually match your sequence, then you can apply it. Like I told you, like also ancient DNA, right? So there are people going back in time in the sediment and finding very ancient DNA, eDNA, or they're doing it in ice. So like the application can be pretty wide, and it's just like very green still. It's quite a new topic, so it's open to research.
Thank you so much, Sophia. So unfortunately, it's time for us to wrap everything up, but we want to thank you, Sophia, again for coming on to share your work with us and giving us a look into the Red Sea Decade Expedition. Thank you to the National Center for Wildlife for their partnership on this expedition. And thank you all to the students and teachers tuning in with us, as well as our interpreter, Dr. Muhammad. Thank you for your excellent questions today. I know Sophia is really excited to hear from other folks interested in science. We're so glad you could be here with us tonight, and we're hosting more live talks with scientists on board OceanXplorer as the expedition continues, and you can find all those details on our social channels where we are at OceanX. Thanks again, everyone, and see you next time.
Thank you. Bye. Thank you, guys.